Thank you very much for this kind introduction and also for inviting me here today to speak in front of all of you and share our recent findings in this project. So in this project we are developing a process to clinically manufacture NK cells that are genetically engineered. So we are using them for allogenic cell therapy. And this is maybe a good example of also the infrastructure in Flemingsberg. So this project is mainly carried out in Evanalicis group in KI. But we are working at the pre-GMP facility where we are currently setting up the process, and the plan is then to move this process to the GMP facility here in Novum at Vecura. So today I'm gonna just give a quick background for allogenic therapies and what the challenges are and how we are planning to approach this, and show some results of our small scale and large scale process development, and then summarize and share some future plans. So just to give some very basic background, if we divide immune cell therapy very broadly, we can divide it into allogenic and autologous therapies. I'll just find my pointer. So when we talk about allogenic therapies, we can receive a donation from a healthy donor, for example, a blood donation where the cells can be retrieved, and they can then be expanded, modified, whatever you want to do, ex vivo, and they are then formulated and can be given to several patients. Whereas when we have an autologous setting, we receive the material from the patient itself that can then undergo expansion, engineering, whatever, and then go to the same patient back. And of course there's pros and cons with either of these modalities. So most of the, for example, CAR T-cell therapies that are on the market today are actually autologous cell therapies. So they are highly personalized and need to be manufactured on demand. One also limitation is the starting material. Maybe the patient is not in a good health state, obviously, so it can be difficult to actually retrieve the cells needed. And there are a lot of logistics and also costs around manufacturing this personalized medicine. And of course the timing is very critical because the patient needs it now, and production processes can take several weeks. On the other hand, allogenic cell therapies have the opportunities to serve several patients from one batch. So there's an opportunity to run large-scale manufacturing, which can also reduce the costs, and also they can be on-demand access or off the shelf, so that the patient, when there's need, can get the medicine immediately. But of course there are also challenges, because in contrast to autologous therapies where the patient has immunological tolerance for the product, allogenic therapies have the difficulty that they can induce a graft versus host disease, and this is mainly for T cells. NK cells, however, are in itself very safe because they don't induce graft versus host disease, but they can be still detected and rejected by the host, so maybe the cells don't last in the patient very long. And this is the challenge that this project is trying to challenge. And there are different mechanisms of how the host can then actually detect and eliminate the cells that you're infusing to the patient. For example, they can detect that the graft cells have a foreign HLA complex, so they have molecules on the surface that can be detected as foreign. And NK cells can also detect alloreactive antibodies and kill the allogenic cell. And there's also missing self-detection because of these different surface molecules. So eventually this graft cell can be eliminated. And there's a lot of research going on of how to circumvent this. And for example, just giving some examples, so for example, you can try to evade this detection by the host cell. If you, for example, remove these surface HLA molecules, or you can eliminate the host immune cells, which of course can be difficult for the patient as well. And the third one, which we are working with, is the disruption of the immunological synapse. So the immunological synapse is needed for one cell to kill the other. And in that itself, there's also several strategies that have come forward. But we are focusing on a molecule that is very present and important in the immunological synapse, and it's called CD45. It's abundantly expressed on all immune cells, and it's a tyrosine phosphatase. And this phosphatase is very important for immune cell activation. It will dephosphorylate certain tyrosine residues and then activate a cascade of activation. And how it then looks, if we have, for example, here a host cell and an allogenic cell, the cell will be detected and CD45 can activate this cell, and it will then be actually excluded from the synapse, which is important in this context also, and the cell can be lysed. And this molecule has been found to be used in nature to actually evade the immunological detection by several viruses, for example, adenovirus and CMV virus. And what these viruses have are proteins that bind to this molecule and thereby reduce its activity and also thereby reduce the function of both T cells but also NK cells. So kind of taking inspiration from this, the group has developed a chimeric molecule, which we then call the CD45 engager. So what it is that the chimeric receptor, kind of similar to a CAR, which has a binding domain that then detects CD45, which is derived from nanobodies. And it also has, which will become relevant later in the talk, it has a little sequence here that we can use for both detection of this molecule on the cell surface. And the theory is then that if we have this allogenic cell that we give to the patient that expresses this molecule, it will still be detected by the host cell. But when the engager binds, this molecule cannot be separated from the synapse, so the synapse cannot form as it should. So the cell is actually spared from being killed by the host cell. And our first proof of concept experiments, we then used a common target cell of NK cells, K562, and we expressed this molecule in these cancer cells, and then we co-cultured them with immune cells, and then monitored their survival and expansion. So in this, I hope it's okay. In this panel we have normal tumor cells, normal K562 cells, and we have several donors of immune cells, and we can see that there's no green cells, whereas when this target cells are alone, they can proliferate and survive. But when the target cells express this engager, we can see that they still persist and expand, so the immune cells are not able to actually kill these cells. So we wanted to use this strategy then to make an allogenic NK cell therapy product. So when we introduce the NK cells to the patients, that they are not eliminated and can survive longer in the patient and have a longer effect. And what we're doing more specifically is to set up a large-scale process to produce these NK cells, expressing this engager, and then for this we're using the Clinimax Prodigy. So here I just have an overview of the process that we are using. So we are starting with leukapheresis products from healthy donors. And these are then, the whole production process is then in the Clinimax Prodigy. So it's a closed system. There's a little picture here on the right. A closed system where the whole process from start to finish happens automatically, and in a closed system, so in a clean environment. And this process is divided into two parts. We are first isolating the NK cells from this leukapheresis, and in a second part we are expanding the NK cells and also transducing them with this construct. And for the second part, we are using a customized application because the process itself doesn't exist as a standard process. So we are activating the cells, transducing them, expanding, and then we want to select for the cells that actually do express the engager on the surface. And then we have another expansion step and finally harvest the cells. But to start with, we needed to set up this process in small scale, because we needed to test the culture conditions, test the transduction, etc. So we started by doing the process in like smaller scale, in normal culture flasks. And what we first wanted to look at is, do we have a good expansion environment for the cells, and do they transduce? Sorry. So first we tested if we can isolate them with this two-step isolation process, which is first a CD3 depletion and then a CD56 enrichment. And this we did on just PBMCs derived from healthy donor buffy coats. And usually NK cells are not so, it's a small population in the blood, so around maybe 10%. And then after the isolation, we get nearly to 100% for most donors. And then we tested our expansion media, and this culture is also feeder cell free. And we can monitor the expansion of the cells, and this here we culture the cells for 14 days, and we saw a strong expansion of the cells. And when we look at the fold changes, we can see that even at the later time points, the cells are still happily proliferating and expanding. So next was to test if we can then introduce this engager to the cells. So for this, we have a lentivirus transduction of primary NK cells, and for those who don't work with NK cells, they are not happily transduced because their natural function is also to fight viruses and virus-infected cells, so they don't like viruses. Anyways, we are trying to make them take up the viruses. So the neat thing about this construct is also that we can measure the expression directly in flow cytometry, because we have this surface marker. So here you can just see some example plots. Here we have the wild type cells, and after the transduction we have around 20% of the cells expressing this molecule. And we test the transduction a week after the transduction to make sure that it's also stable. And we found that even with this low MOI of 2, we can get a transduction rate of around 20-25%. So this sequence that we are using to detect the cells, we also want to use the same sequence to actually enrich this population, because we would like to have a product that has a high percentage of these cells, because 20% would maybe be a bit little. So we are using this molecule to enrich the cells using magnetic isolation. And this step we also first optimized in a smaller scale, and we actually used a different machine for that, which has some similarities to the Prodigy, but it's called the Automax. So what it can do is can label the cells automatically. It's like magnetic beads that are coated with antibodies, so they can bind to the target, and they can then go through like a column where the labeled cells get stuck and the unlabeled cells fall through, and then you can retrieve the cells that are retained in the column. So we used this system because it gave us a bit more flexibility to test a lot of different conditions. What we tested is different speeds in what the cells will flow through the column, because this can change both the recovery and the purity of the cells. So we used our standard process where the NK cells are isolated, activated, transduced, and then 1 week later we add this additional step where we isolate them using these magnets. And so what we can see here is again the wild type cells, and then there's the ones that are transduced before the isolation. In this case it was a bit higher transduction, and then we can retrieve both the positive and the negative fraction of this magnet, and we can see that we have a highly enriched population here in the positive fraction, and we have some cells that are expressing the molecule, but they are maybe not expressing it very strongly, so they also fall into the negative fraction, but it's only a small percentage. And what we found is that, I'm not showing it in more detail here, but what we found is that the fast flow through gave a very high purity in this setting, but we had very low recovery, so we need to find a bit of a balance, because we also need, we want to of course achieve high yields for the clinical production. So we found that actually slower speeds would maybe lead to a tiny bit lower purity, but significantly higher recovery. So the idea is that we can use these data as like a starting point when we move to the Clinimax Prodigy, because it's less flexible to test a lot of different variables. And this also brings me then to the last part. So now that we set up the process and the culture conditions and the transduction, we were ready to then move to the large scale in the Prodigy. And for this experiment, we received leukapheresis from a healthy donor. And the first step, as I said, was then to isolate the NK cells in this two-step process. So in the left graph, we can see the starting material, and this little quadrant are the NK cells, so we have around 7-8%, so that's quite common. And then after this isolation, which happens completely automatically, we got a very pure NK cell population, and we could then start the NK cell culture with the high NK cell number, which we would not be able to retrieve from, for example, a normal blood donation. And then we also did a transduction. This also happens automatically in the machine, and it's a spinoculation. So the machine has like the culture chamber itself is a centrifuge. So the cells are transduced in that way. So we transduced the cells, and then we took a sample after a week, and we could see that we got similar transduction rates in the Prodigy compared to our small scale. So this one we ran in parallel just to have a reference from previous experiments. And the transduction rates were similar, and we monitored them over the whole culture, and in the Prodigy was a pinch higher, but let's say they were very comparable. And the last parameter, of course, that is very important is the expansion rate of the cells. So here we also compared with the Prodigy and the small scale, And the kinetics seem to be very similar, even though we get different numbers, of course. But if we look at the fold change, we can see that they overlap quite well. So it was really possible to translate this small scale process to the large scale and get comparable results. So that brings me to my summary. So we managed to set this process up in small scale, and we also could establish this isolation step, and then we were successful to actually move this to a larger scale in the prodigy. Now we're just starting with these experiments, so this will be what will be going on from now on, because soon we will also implement this additional application that we can then introduce this additional isolation step into the automated process in the prodigy. So that should be coming soon. And then, of course, the end goal is to transfer this to the GMP facility here at Vecura and prepare for a clinical study. So with this, I would like to thank all of you for your attention, and also thank everyone in the team, even Alici's group, and also at Vecura, and the collaborators, funding bodies, and of course the donors that give us the material. And thank you for your attention.